Can you share your screen? Yeah.
Okay. Can you all see my screen and hear me well?
Yeah, we cannot hear you.
Okay. Now, can you hear me now?
Yeah, it's just not very loud, but it's okay.
Okay. So first, thank you so much for the very nice introduction. First of all, I'd like to thank Open Box Science for giving me this great opportunity and all of you for joining us today. So today I will be sharing our recent finding on a transcription mechanism that regulates regional heterogeneity of astrocytes and also their functions. So as we've learned from Eddie and Kevin, astrocytes play very diverse functions in the brain, including regulating synaptic activity, promoting synaptogenesis, providing energy support, and also regulating blood-brain barrier. And because of that reason, it's not surprising that they are involved in a great number of CNS injuries, as we just learned from Kevin, and also a lot of other.
Anna, sorry, can I please ask you to be a bit closer to the microphone because we cannot hear you very well?
Okay, it's now better?
Not really. Yeah, I don't know.
Let me see if I can fix by changing the setting.
Yeah, it seems better now.
Okay.
Oh no, I don't know. Try to.
So I'll just speak louder. It's
yeah.
Now better? Okay. So
yeah.
Talking about that, astrocytes is involved in a great number of different CNS injuries and neurological diseases. And these diverse functions in the brain raise a question of whether there are differences of astrocytes. And indeed, recent studies has shown that astrocytes from distinct regions of the brain are very different. Molecularly, they show differences in their transcriptome, proteome, and also their metabolite contents. And morphologically, they show differences in their complexity and their territory sizes, and also their distance to the synapse. Functionally, they show differences in their ability to support neuron and also their calcium activity. So together, this evidence shows that clearly there is a regional heterogeneity among astrocytes. However, we still don't know what drives this regional difference. To find the answer, we looked into studies on developing CNAs, where we've learned that transcription factors actually play critical roles on regional allocation of astrocytes. For example, in the developing forebrain, different transcription factors are expressed in different segments of the ventricular zone. And these different segments then give rise to astrocytes that locate in the respective brain regions. In other words, this regional information is encoded by transcription factors. So knowing this, we were really curious about whether transcription factors also control regional identity of these astrocytes in the adult brain. So to find out, we use our long-standing interest, transcription factor NF1A as an entry point. Based on previous studies from the lab of David Anderson and us, we've learned that NF1A is upregulated and is required for astrocyte generation during development. But we recently also found that even after this generation phase, the expression of NF1A does not go down, but it continues to express in mature astrocytes. This then raises a question of what is the function of NF1A in these mature astrocytes. And we suspect that these mature astrocytes must repurpose NF1A and use it to regulate other functions. So to be more specific, here we are asking two questions. First, whether NF1A regulates regional heterogeneity of these mature astrocytes in the adult brain. And second, whether NFI regulates physiological function of these mature astrocytes. And here we will be focused on the communication between astrocytes and synapse or synaptic regulation. So to find out, we generated a conditional knockout system which allow us to specifically delete NF1A in the mature astrocytes. And we did this by crossing astrocyte-specific LDH1, L1 pre-ER with NF1 flux allele. And also we performed this tamoxifen induction at 5-week-old when astrocytes are already matured. We then waited for 2 or 4 months before we analyzed multiple brain regions, including olfactory bulb, cortex, hippocampus, and brainstem. So first thing after we generated these mouse, we validated the system using immunostaining. And here we found 2 things. First, NF1A is equally expressed in 80% of astrocytes across all these regions. And second, the knockout efficiency was consistent across regions. So knowing that this system is robust, we then want to find out whether this loss of NF1A caused any changes in astrocytes, including their morphology, calcium activity, behavior, as well as transcriptome. So to look into morphology, we use astrocyte reporter ALDH1, L1 GFP to label these astrocytes and perform show analysis to quantify their morphological complexity. And to our surprise, we found that removing NF1A from all these brain regions did not cause any changes in olfactory bulb or brainstem. However, we did notice there is a reduction of the branch length and the morphological complexity in the hippocampus, indicating that there is a region-specific dependency. So knowing this, we next want to find out whether this region-specific dependency also applies to calcium activity. To look into astrocytic calcium, we express in this astrocyte GCAM6 and apply 2-photon light imaging in this brain slice. And here we found that loss of NF1A did not change calcium activity in the olfactory bulb, cortex, or brainstem. However, it causes a reduction of calcium activity in the hippocampus, as demonstrated by reduced amplitude and frequency. So this shows that this regional dependency also applies to calcium activity. So if activity shows this region-specific reduction, what about behaviors? So to find out, we perform behavioral tests corresponding to each brain regions. In the olfactory bulb, we found no change in their odor detection. In the cortex, we found no changes in their social behaviors. And in the brainstem, we also found no changes in their breathing behaviors. However, in the hippocampus, we found a significant reduction of their spatial memory, as demonstrated by reduced contextual fear and novel place recognition. So this again shows that this region-specific dependency also applies to behavioral label. So to summarize the findings so far, we found that NF1A, although NF1 is expressed in astrocytes of all these regions, removing all of them most dramatically caused changes in the hippocampus, as demonstrated by reduced morphology, calcium activity, and learning and memory deficit. And this really caught our attention because it's almost like NF1A expressed in these other brain regions are just being expressed and doing nothing. And we thought it was really odd, and we are curious about how does that happen. And because NF1 is a transcription factor, we next want to find out whether NF1 preferentially regulate genes in the hippocampus, which then cause this region-specific dependency. To find out, we perform RNA sequencing to compare gene expression with and without NF1A. And shown here are volcano plots displaying upregulated genes in red and downregulated genes in blue. And each of these dots represent one gene. So first thing we noticed was that loss of NFIA caused way more genes being dysregulated in the hippocampus compared to other regions. And from quantification, you can see that the difference is more than threefold. So this indicates that NFIA preferentially regulate genes in the hippocampus. Moreover, from our principal component analysis, we found that indeed, astrocytes of distinct brain regions has their own molecular signature. And removing NFIA does not change too much of that in the cortex, olfactory bulb, or brainstem. However, in the hippocampus, loss of NFIA did result in a loss of hippocampal specific signature. And these NFIA deficient hippocampal astrocytes become more like cortical astrocytes. So these findings indicate that NFIA is required for maintaining the hippocampal specific signature. So now we know that NFIA preferentially regulate genes in the hippocampus, and such regulation is required for maintaining hippocampal specific signature. Knowing this, we next want to find out what mediates this region-specific gene regulation. And we suspect that NFIA might bind to gene differently across region, which then cause this region-specific gene regulation. So to find out, we perform chip sequencing, which allow us to identify the binding targets of NFIA and analyze them. And here we choose two brain regions, but the hippocampus and the olfactory bulb, which represent regions that is regulated and not regulated by NFIA. So then we perform chip sequencing. And we found that in the hippocampus, NFIA binds to more than 1,700 of genes, but in the olfactory bulb, it only binds to 75. This indicates that NFIA binds to genes in the hippocampus, but not in the olfactory bulb. And this finding, this differential gene chromatin binding might explain why NFIA preferentially regulate genes in the hippocampus. But it also raised another question about what caused this differential gene binding. And our hypothesis was that NFIA may either associate with another cofactor that inhibits its binding to the chromatin in the olfactory bulb, or facilitate its binding in the hippocampus. To find out, we perform IP mass spec, which allow us to identify the associating partner of NFIA. And here we found that in the olfactory bulb, NFIA preferentially interact with another transcription factor called NF1B, which belongs to the same transcription factor family. And we also validated this finding by immunoprecipitation, showing here that there are more interaction in the olfactory bulb compared to hippocampus. So now we know that they interact with each other in the olfactory bulb, but we still don't know whether this interaction really inhibits the binding of NFIA to its target. And to test that, we need to remove NF1A and see whether that will, we need to remove NF1B, excuse me, and see whether that will increase NF1A binding. So to find out, we deliver NF1B essential RNA into the olfactory bulb by using AAV virus, which causes a reduction of NF1B expression. After that, we then perform again NF1A chip sequencing. And here we found that removing NF1B dramatically increased the binding by NF1A, and the number of gene bound jumped from 22 to more than 700. So this indicates that the association between NF1A and NF1B in the olfactory bulb actually inhibit the binding of NF1A to their targets and the subsequent gene expression. So to summarize the findings of the second part, we found that NFIA preferentially regulates genes in the hippocampus, and that is correlated with its region-specific gene chromatin binding in the hippocampus. And in the olfactory, NF1A and NF1B associate with each other, which inhibit the binding of NF1A to its targets. And because of that, removing both NF1A in these respective in both brain regions only affect the hippocampal astrocytes. So having identified this region-specific gene transcription regulation, we next want to focus on the hippocampus. And here we want to find out how does NFIA regulate the function of astrocytes in this region. Specifically, we want to find out how does it regulate the crosstalk between astrocytes and the synapse, and which then results in this learning and memory deficit. To find out, we went back to our morphological analysis, and from here, we suspected that these stunted astrocytes might not be able to communicate with the synapse because they are simply far away from the synapse. So our next question becomes whether proximity between astrocyte and the synapse is altered. To do so, we used a Fred, to look into the proximity, we use a Fred-based optical method called neural astrocyte proximity assay, which allows us to look into measure the distance between CA3 axon projection and CA1 astrocytes. And here we found that NF1 deficient astrocytes has reduced number of red signal, indicating that the distance between this NF1 deficient astrocyte and the synapse has become longer. So if the distance becomes longer, can these astrocytes still listen or detect neurotransmitters? And to look into neurotransmitter detection, here we focus on the glutamate. And to measure that, we virally express glutamate sensor into CN1 astrocytes and apply neuronal stimulation and look into their response. And here we found that NF1 deficient astrocytes cannot detect neurotransmitter in response to neuronal stimulation, indicating that this astrocyte can no longer listen to neuronal signal. So if that's the case, what about downstream calcium activity? To look calcium activity, we express GCAM in these astrocytes and again apply neuronal stimulation. And again, we found that their calcium activity was impaired, indicating that the communication between astrocytes and synapse was impaired. So if the communication becomes inefficient, what would that change neuronal function? And to look into neuronal function, here we measure long-term potentiation because that's a cellular correlate of learning and memory. And after we apply stimulation in Sheffer collateral, we found that the LTP in the NF1 knockout animal is totally inhibited, indicating that astrocytic NF1A is actually required for hippocampal neuron to establish their LTP. So knowing this, we next want to find out what causes this impaired LTP. To know that, we need to look into what happened between astrocytic calcium and neuronal LTP. So based on previous studies, we've known that D-serine is released from astrocytes and is triggered by astrocytic calcium. And D-serine is an NMDA receptor agonist, which facilitates LTP induction. So here we will be testing whether D-serine and calcium are involved in this process. So to test whether D-serine is required, here we supplement D-serine right before neuronal stimulation. and measure long-term potentiation in control and knockout animals. And we found that these theory rescued this LTP impairment, indicating that it is the missing component that underlying this LTP impairment. Knowing this, we next want to find out whether calcium is also involved. And to do so, we restore calcium in CL1 astrocytes by overexpress GQ drug and in combination with CMO treatment on the brain slice. And again, we found that restoring calcium rescued LTP impairment. So together, this data suggests that deserine and astrocytic calcium is the missing part that explains this impaired LTP. So together here, we have shown that in the hippocampus, loss of NFI results in reduced neural astrocyte synapse proximity, which then leads to reduced glutamate detection and calcium activity. And this then in turn cause a reduced amount of deserine that can reach to NMDA receptor and the subsequent LTP impairment and learning and memory deficit. So overall, there are two major points in this study. First, we found that NF1A regulate genes in the hippocampus has this region-specific gene regulation, and it preferentially regulates genes and binds to genes in the hippocampus. And that was partly due to region-specific association between NF1A and NF1B in the olfactory bulb, which inhibits its binding to the targets. And because of that reason, removing NF1A will only cause changes in the hippocampal astrocytes. And the second point in this study was that we identified a transcription factor, in this case NF1A, that is involved in this astrocyte synapse communication. And in the hippocampus, loss of NF1A causes shorter branches and reduce calcium activity, which then impair the communication between these two components and reduce LTP and learning and memory deficit. So going back to our original question, in these studies, we have identified transcription factors, in this case, NF1A, that regulate astrocyte synapse communication, which has never been shown before. And in the future, it is important to identify what are the downstream target genes that is responsible for these functions. And moreover, by showing that a transcription factor could define hippocampal specific signature, we have identified a transcription mechanism that controls regional heterogeneity. And in the future, it will be interesting to find out whether there are other transcription factors that are responsible for astrocytes of other brain regions. And one thing I would like to emphasize is that the expression pattern of a transcription factor does not necessarily match with its transcription activity. For example, NF1 is expressed everywhere, but it only regulates genes in the hippocampus. And in the future, it will be helpful to bear that in mind when looking to other candidates that regulate the heterogeneity of astrocytes. And with that, I'd like to thank my lab, especially my mentor, Dr. Benjamin Denning, who has been giving me great advice since day one. And also Dr. Debo Sarda for helping me with biochemical experiments, and Dr. Jinsen Wu for helping me with the electrophysiological experiments, our collaborators, my committee members, and our funding sources. And with that, I'd like to take questions.
Thank you, Anna, for the nice talk. So here we are. How do you distinguish between effects on NFA1 on gene expression in astrocytes versus neuron in these bulk RNA chip-seq experiments?
Wait, can you say that again? I lost you for a second.
Yeah, it's basically how do you distinguish the effects of NFA1 NFIA on gene expression in astrocytes versus neurons in the bulk RNA and the chip-seq? Because I guess you did bulk, or you divide them, you sort them.
Yeah, so great question, thank you.
Yeah.
So first of all, ours, can you still hear me? Okay, first of all, our RNA sequencing was done from sorted astrocytes, so it's astrocyte specific. And second, for chip, we did bulk, but astrocytes majorly, NFI is majorly expressed in astrocytes. So that's kind of enrichment of the astrocytic chromatin signature by pulling down NF1A. So that's our method to distinguish between astrocytic NF1A and neuronal NF1A.
Great work, Anna. There are two questions here from the same person. He says, beautiful work. When removing NF1B in olfactory bulb, did you see the astrocytic signature become more like hippocampal astrocytes? And the second question is, I'm curious if for those NF1A CKO hippocampal astrocytes, do they increase the interaction to cortical neurons since their genetic signature is more like cortical astrocytes?
Thank you for great question again. So to the first question, we did not look into the astrocyte after the histological signature of astrocyte after we knocking down NF1B. But that's a very interesting thing, and currently we have a graduate student that is working on that. And for the second question, the question is to more impact.
Anna, if you want, you can stop sharing the screen and you can also read the questions. It's easier if you want in the chat. Okay. If it's easier for you.
So
it's from Jimmy Min.
Okay, yeah. So for the second question, we did not find any more close interaction because these astrocytes still in the hippocampus.
Yeah. And we have one more question. So is this arena good clinical therapeutic for impaired LTP or patient with similar symptoms? Nice talk.
Yeah, definitely. I think that's very promising. But we really need to test on a clinical patient to know whether that is really effective on human. So thanks for the question, but we don't have an answer yet.
Yeah, thank you, Anna, for the very nice talk.
Thank you. Thank you for